Hello, it's an honor to be with you here today at this outstanding virtual meeting hosted by LabRoots. And this has been a banner year for CAR T-cell therapy, particularly in solid tumors. And we're finally, I think, turning the corner. So in this talk, I'll synthesize for you the recurring themes that I believe underlie clinically potent CAR T-cell products. So those where we're at least getting efficacy signals, and also highlight the common challenges that remain and how we can address them. Here are my disclosures. So as you can see here, solid tumor CAR T therapy faces four major barriers. This isn't an exhaustive list, but let's just bucket it like this for now. First, antigen constraints and escape. There are a few truly tumor-exclusive antigens. There's low and heterogeneous antigen expression and antigen loss variants that can blunt the post-infusion proliferative burst needed for CAR T cells to reach a therapeutic level of expansion. Secondly, and I have this up here, suboptimal in vivo expansion. The CAR T cell counts in these patients with solid tumor indications are often 5 to 10-fold lower than in blood cancers if you're administering these therapies systemically, and that limits the initial tumor clearance. So unfortunately, it's sort of a failure right out of the gate. Third, there could be impaired trafficking and homing, and this could be attributed to chemokine receptor mismatches, the abnormal tumor vasculature, and also the dense stroma that often prevent sufficient CAR T cell recruitment into the tumor bed. And finally, if your cell product can infiltrate into the tumor bed, there's limited persistence and early hypofunction that's been seen in many trials, and that's because of multiple immunosuppressive axes that are operative within the tumor microenvironment that drive rapid exhaustion and other sorts of defects, and CAR T cells ultimately fail to establish a durable tissue-resident population. So on the right, you can see how intrinsic qualities and also extrinsic tumor barriers really interact to create these challenges, and that sets the stage for both engineering and manufacturing solutions that I'll explore. On this slide, we see some clinical hallmarks shared by the most potent CAR T products that have been tested in solid tumors that have been validated clinically as well. So first, as you'll see here, high antigen density and patient selection. So studies enrolling only patients with at least 40% target positive tumors, such as the CT014 study of a Claudin 18.2 directed CAR T cell product. This can markedly improve response rates. Second, administration of high doses of CAR T cells and repeated infusions, administering up to 5 times 10 to the 8th CAR T cells with retreatment of patients as needed tends to correlate with more durable regressions and may even reduce reliance on initial aggressive lymphodepleting conditioning prior to adoptive transfer. Third, robust in vivo expansion and detectable persistence. And so in these trials where, again, we see some anti-tumor potency signals and, dare I say it, even efficacy, responders exhibit peak CAR T levels above 10 to the 4th copies per microgram of DNA, and then maintain these circulating cells for weeks to months. Fourth, local delivery. So if you can't, it is somewhat of a numbers game, and if you can't get an adequate number of your engineered T cells to the tumor site, if the indication permits, intratumoral or regional infusion can overcome, some of these trafficking barriers and certainly increase, the number of CAR T cells locally where they need to be. And fifth, armored constructs and microenvironment modulation is very important. So CAR T cells, for example, engineered to secrete cytokines or to resist suppressive cues, have produced deeper regressions, at least in early phase studies. And finally, manageable safety profiles. So careful management of CRS, ICANS, ICHS that has now come to light, allows for more aggressive dosing with tolerable toxicity. So really the goal here is to decouple the anti-tumor potency from these hyperinflammatory toxicities. So these features I feel define the blueprint really for next generation solid tumor CAR T strategies. Next, I'd like to present a few clinical examples that really highlight why robust expansion and durable persistence, even in the solid tumor setting, are critical for successful outcomes with CAR T-cell therapy products. And although this might seem very evident, it remains an area of active debate. Do we need persistence? Maybe we only need persistence in certain indications. So as a good example, I wanted to present to you a mixture of both published and unpublished data with our TGF-beta resistant prostate-specific membrane antigen-directed CAR T product in metastatic castrate-resistant prostate cancer. So the expansion of CAR T cells in the peripheral blood is shown on the top right. And in this trial, there was a clear dose and lymphodepletion dependent effect on the degree of expansion of the product and in turn the anti-tumor activity, at least to some extent. And so here's unpublished data from down on the left most corner of the slide from a more recent patient treated in our high dose cohort, showing robust expansion of the armored CAR T cells in the peripheral blood, coincident with the precipitous decline, as you can see, in serum PSA levels, which we used in this study as a surrogate marker for tumor burden. And accordingly, this patient exhibited a high CAR marking in tumor tissues, that's shown in the bottom right, which was associated with at least transient tumor shrinkage. So obviously, to some extent, again, it's a numbers game, and if we can get into the right therapeutic window, we have a shot at achieving perhaps clinically meaningful tumor regression. So unfortunately, though, as I alluded to previously, this more recent response was accompanied by ICHS in the subject. So, the challenge, I think, with most solid tumor CAR T products is, again, decoupling that anti-tumor potency with this sort of toxicity, which is becoming more and more apparent in the field. However, in some cases, both systemic and intratumoral or regional CAR T expansion remain inadequate, and we've observed this in our mesothelin-directed CAR T cell trial for pancreatic cancer, and this was recently published in Cell Reports Medicine. And so, as you can see here, regardless of the administration route, peripheral blood monitoring and tumor biopsies really revealed minimal CAR T proliferation, persistence, or bioactivity, as you can see here. And so without obviously achieving a therapeutic threshold for CAR T expansion, meaningful tumor regressions in this setting are highly unlikely. So recently, my laboratory set out to solve this, hopefully to some extent, with a vaccine-like strategy. And this is some more unpublished data where we created a bridge sort of molecule in which we have an affinity-tuned anti-CD19 single-chain variable fragment on one end of the molecule, and then on the other end of this bispecific molecule, a protein fragment that bears the epitope that the solid tumor CAR T cell product recognizes. In this case, just to follow the clinical data from the previous slide I showed you, we used anti-mesothelin CAR T cells. And the idea here is to really ride that B cell wave, and remember that B cells are semi-professional antigen-presenting cells, and so it might be possible to get additional co-stimulation with this strategy, in addition to really spurring on antigen dependent expansion in the blood. So here in the middle, you can see that by spiking in this bridge protein in an in vitro culture system containing our mesothelin-directed CAR T cells, and B cells, we can potentiate CAR T proliferation in a dose-dependent manner. And as a positive control, we co-expressed the same anti-CD19 CAR, but unlike that strategy, there's markedly less collateral damage to the B cells, as you could see here, again on the right. And we have much more in vitro data that I don't have time to show you, but to put this to the test, we used a humanized mouse model in which NSG mice were infused with stem cells, followed by tumor, and after B cell reconstitution, we infused untransduced, or again, our anti-mesothelin CAR T cells, followed by a weekly boost with the bridge protein. And as you can see here on the bottom left, there was a significant increase in circulating CAR T cell counts using the bridge vaccine with minimal collateral damage to B cells, as shown here. And that increased expansion capacity was associated with better tumor control compared to the CAR T cell monotherapy. So as you would expect, this coincided with improved tumor infiltration when you use the bridge protein, and that's exemplified here in this IHC staining for human CD3 within the tumor xenografts. So this can, this technology we think can apply to all sorts of CAR T cell products, whether you're microdosing with ex vivo manufactured products or even generating CAR T cell products in vivo. This has a lot of translational potential and clinical utility, I think. So what is another way to overcome issues with expansion and persistence while simultaneously addressing antigen heterogeneity? And that's what I want to discuss with you in these next slides. Well, if the tumor is accessible and you can safely re-administer cells, as I mentioned, locoregional or intratumoral delivery becomes an attractive strategy. And so this year has really brought encouraging results in this regard from groups here at Penn, City of Hope, and Massachusetts General Hospital using this approach. And so I'll speak for our study. So in our open trial, we're testing a bivalent CAR T cell product targeting the epidermal growth factor receptor and the IL-13 receptor alpha 2 via intrathecal delivery of the product. And of course, that bypasses the systemic circulation and really deposits the cells directly into the CSF. And so by focusing on EGFR and IL-13 receptor simultaneously, we can enhance tumor targeting breadth really for aggressive multifocal disease. And so this, as you can see, is a 3+3 dose escalation design, and enrolling patients with EGFR amplified recurrent glioblastoma, again delivering CAR T cells via an Ommaya reservoir. And so our primary endpoints are dose-limiting toxicities and, of course, safety profiling, and we have some secondary and exploratory analyses that include CAR T pharmacokinetics in both the spinal fluid and blood, cytokine measurements, and histologic assessment of CAR T infiltration in the tumor tissue. And so, again, this locoregional bivalent CAR strategy aims to maximize on-site potency while minimizing some of the off-target toxicity that we previously discussed, which is represents a critical balance for effective solid tumor CAR T therapy. So just to show you some of the preliminary data, which is now published in Nature Medicine, on the left you could see CSF fluid CAR copy kinetics from 3 of our initial patients who were treated. So these individuals achieved peak CAR T cell expansion in the CSF by day 7, and detectable persistence, detectable CAR marking through day 28, and that really indicates successful on-target delivery and survival in the central nervous system. And clinically, 8 of 13 evaluable patients now, so 62%, experienced tumor regressions by modified RANO criteria, including even 1 confirmed partial response. 1 patient also achieved durable, stable disease lasting beyond 16 months, And I think that that highlights that with a strategy like this, there is potential for long-term control even in this very aggressive disease. And so, these findings, I think, provide at least proof of concept that locoregional delivery of a CAR T product targeting at least two antigens can really achieve robust expansion, persistence, and preliminary signals of efficacy while minimizing systemic exposure. That's been an issue. So I wanted to close today with an exciting yet straightforward manufacturing innovation out of my lab that has the potential, we feel, to transform solid tumor CAR T therapy and can really be adopted by any CAR T program with minimal disruption. So I wanted to give you all something to take home with you to your own centers, for your own programs. And I just wanted to say that we've advanced this approach into the clinic, and we've just dosed our first 2 patients with CAR T cells manufactured in this manner. And while it's too early to report clinical outcomes, the preclinical data are compelling, and I hope these results will again encourage other groups in the field to incorporate this modification and move us all closer to durable, hopefully, cures for solid tumors. So as you can see here, we recently developed a strategy that really transforms peripheral blood T cells into tissue-resident memory CAR T cells. And these cells are engineered to have superior trafficking capabilities and enhance effector functions while specifically targeting tumor tissues by virtue of a chimeric antigen receptor. Their unique ability, when programmed this way, allows them to localize and persist in the relevant tissue compartments without recirculating. And the goal here was to provide sustained anti-tumor activity. So to do this, we optimized culture conditions for manufacturing CAR T cells in the presence of low levels of recombinant human TGF-beta. So as shown on the slide, CAR T cells exposed to TGF-beta, what we're calling CAR tissue-resident memory cells, or CAR TMs, upregulate several classical markers that are, associated with the TRM phenotype, including CD103, CD39, and CD49A. And these CAR TRM cells show enhanced production of multiple effector cytokines and a comparable level of ex vivo expansion during production, implying that proliferative capacity initially and certain T cell effector functions are not compromised by the inclusion of TGF-beta during the expansion process. Remember, it's low-level TGF-beta, which is why we're seeing this effect. So as you saw on the previous slide, flow cytometric analysis of our CAR tissue-resident memory cells reveals the acquisition of a resident memory phenotype imparted through TGF-beta signaling. So to expand this finding, we really sought to obtain a comprehensive view of the transcriptional landscape of these CAR TRM cells by initially performing bulk RNA sequencing. And so principal component analysis of the transcriptomic data indicated that TGF-beta exposure was really the strongest driver of transcriptional variants across multiple donors. And consistent with our flow cytometry data, these CAR TRM cells were significantly enriched in CD69 positive, CD103 positive, CD8 positive resident memory T cell signatures, and they also exhibited reciprocally reduced non-resident memory T cell transcriptional profiles. And just to get a little more specific there, the CAR TRM cells expressed high levels of these memory associated genes, encoding cell surface molecules and transcription factor genes like EGR2 and transcription factors within the NR4A family, while downregulating KLF2, which is of course essential for T cell recirculation and inhibition of the differentiation of TRM cells. And so this increased resident memory transcriptional program in CAR TRM cells may be also partly attributed to hypoxia pathway upregulation, which we also saw in our data, and that's known to further facilitate resident memory T cell differentiation. And that sort of indicated to us, although we haven't experimentally validated this, a possible overlap between TGF-beta signaling and responses to low oxygen tension. And I just wanted to mention that both CD69 and CD103 are relevant to the survival of tissue-resident memory cells. So 103 obviously allows the cells to tether and hang on to the epithelial tissue, and CD69 has a relationship with S1P, which allows the T cells to shut down tissue egress by the transcription factor KLF2 that I mentioned. Just to reiterate, we obtained those marked transcriptional changes due to the production of our CAR T cells with low levels of TGF-beta, but really we wondered if these differentiation states were transient or perhaps epigenetically or more permanently enforced. And so we worked with Shelley Berger's lab, again here, she's a great neighbor at the University of Pennsylvania, to conduct these analyses. So we investigated in this vein whether transient exposure to TGF-beta during CAR T cell production can induce epigenetic rewiring of CAR TRM cells to sustain the acquired stem-like resident memory phenotype. And ATAC-seq analysis that you can see here shows that CAR TRM differentiation really induces global chromatin remodeling and significantly increases chromatin accessibility at some of the resident memory gene loci that we previously discussed, while decreasing accessibility at non-resident memory genes. So peaks here are enriched in stemness-related gene regions such as TCF7 and the IL-7 receptor encoding gene in these CAR TRM cells. Okay, so let's put this programming to the test. So in vivo studies showed that M5 anti-mesothelin CAR TRM cells exhibit remarkable tumor control in comparison to conventionally expanded CAR T cells. And these studies were done in 2 different pancreatic tumor xenografts that are tried and true. And improved anti-tumor activity was due to a significant increase in the number of CAR TRM cells within tumors. So you're looking at the TIL infiltration here, which is partly due to improved early localization of the T cells to the tumor. And I don't have time to show you this data, but we did conduct these studies. So we saw that both T conventional and tissue resident memory CAR T cells are highly enriched within these CD103 positive, CD39 positive TILs, but mice treated with CAR TRM cells show a significant increase in the absolute number of tumor infiltrating 103 positive, CD39 positive cells. And that indicates to us that the TGF-beta induced stem-like resident memory population can give rise to a large number of resident memory CAR TILs. We also saw in our CAR TRM population increased frequencies of cells co-expressing inhibitory receptors, which often indicates that the cells may be exhausted. Certainly these cells aren't exhausted, but it's interesting that the low-level TGF-beta induced programming of these cells enforced this state that was in part marked by, again, the co-expression of PD1 and TIM3 or PD1 and LAG3. So again, they're not exhausted, but they sort of look like TILs that you would find in the tumor microenvironment, and we're still studying why that is. Just to conclude, our collaborative efforts show that multimodal engineering strategies are essential to overcome the 4 geographic barriers of solid tumor CAR T therapy that I talked about in this lecture. Those are expansion, persistence, trafficking, and functional potency. I showed you that regional delivery techniques such as intrathecal and intratumoral infusions may bypass homing obstacles and limit off-target effects to some extent. Armored CAR T cells and multi-antigen targeting can address tumor heterogeneity and antigen escape. And our bridge protein vaccine provides a powerful targeted boost to in vivo CAR T expansion. Further, memory-like and tissue-resident programming sustains CAR T persistence and effector function within the tumor microenvironment. And finally, by closing the loop in what I like to think of as an iterative bench to bedside to bench cycle, we can hopefully refine these solutions and accelerate durable clinical success in solid tumor CAR T therapy. And I just wanted to close by acknowledging many of the folks who contributed to these studies here at Penn and the Center for Cellular Immunotherapies or in other divisions, and of course all of our wonderful funding sources. Thanks.